Welcome back. I want to start informing everybody that I would want to inject some puns in my discussion. I received this suggestion that I should not be too serious in the way I present my videos, but there's nothing I could do. I could not see anybody except my computer, except to really seriously let you know exactly what's in my mind. So I'm going to start injecting puns in my discussion, which I hope will not sound corny. Really? I got this gut feeling that my desire to be creatively funny will not end well. But we've been very seriously talking about the human body, and I thought yeah, I'd heed to the suggestion, start talking about the digestive system one bite at a time so the information gets easier to digest. And pretty much, I know I have this gut feeling, and my stomach is always up for a little food for thought. I hope yours will be as well. So let's get ready. Let's start. We are now on the first part, and I want to let you know that chapter 23, which is the digestive system, is made up of three videos, and they're all long, and I shortened each of them, all of them. But I would like to go through all of them really very briefly. I added a little bit more uh, pictures and diagrams so I could effectively discuss them. So, what are the functions of the digestive system? First, your digestive system allows food to get into the body. And then the food just don't stay there as food. We have to make sure the reason why we take food is so that it can nourish us. So it breaks it down to nutrient molecules, smallest as possible, that could be taken in, absorbed. And then we have the capacity to absorb these broken down molecules into the bloodstream. And then pretty much like get rid, another part will be to get rid of any undigested remains of the food that we have taken. And it's each of this each of these functions are actually divided by several organs that are highly specialized in doing their work. The mouth, for instance, takes in food and it has the ability to break down food particles and assist in pretty much like starting in its simple chemical digestion because the saliva has some chemicals on it, and then the food now gets into the is swallowed in and it goes right through the pharynx all the way into the esophagus and to the stomach. But that thing of the movement of swallowing it is not going to be possible had not been for the production of saliva, which is a production of salivary glands. And we have several salivary glands, which I will be discussing to you later. The esophagus, which is actually the tube that connects the oral cavity to the stomach, pretty much allows the transport of food from the oral cavity down into the stomach. The stomach would be chemically as well as mechanically digesting food, and the main part in the stomach is the production of an acidic compound which you call the hydrochloric acid. It blends well with the food and it helps in the production of a special type of enzyme, or rather the activation of a special type of enzyme which we call pepsine from a from its precursor which called pepsinogen. So, with the help of this hydrochloric acid, pepsinogen gets converted into pepsine, and pepsine is a protease. It is actually the enzyme that digests protein. So that's actually the initial digestion in the stomach, among other things that brings about the churning and the movement of the stomach and the way our stomach would behave once food is inside it. Pretty much is the function of many different types of cells, which we are going to be discussing when we get there. Then food now is going to be transported little by little by via an opening here, which is your gastroesophageal sphincter, and would bring food, which now we will call a chyme, uh, once it reaches the intestine, or once the chyme has actually like been um, once the chyme gets in contact with the gastric juice here in the stomach, pretty much. Um, okay, let's first of all let me just mention why I'm saying chyme, and that's actually the food. Well, first of all, let me introduce to you the language or the different ways by which we call food and why we call it different names once it gets through the different parts of the gastrointestinal tract. Food, when it actually gets in contact with saliva, you call it bolus. Okay, so saliva plus food, and you pretty much masticate it or grind it in your mouth. You chew on it, it becomes bolus. So you swallow the bolus. The bolus now gets in contact with the gastric juice, um, um, the acidic mixture of hydrochloric acid and mucus and other enzymes that's in the stomach, plus your bolus will now be called chyme. Now, the chyme is actually the name that it is going to uh, be holding on all the way to its travel to the small intestine into the large intestine until it becomes really ready to be eliminated outside, where we call the chyme the feces or the fecal matter. Okay, so the food that we take in would not stay as food the way we know it inside our stomach because of its encounter with many enzymes and with many products of digestion. So now the chyme, let's go back to the stomach where it has the chyme. The food now gets into the small intestine, and the small intestine it walks it actually going to be acted upon by secretions coming from the liver, which is actually stored in the gallbladder, which we call bile. If it is fatty, then you know you are going to be the food is going to be acted upon by this bile, and um, the enzymes also of the pancreatic juice is going to be acting on it, and we'll be discussing the different enzymatic action that happens right through the small intestine. And once digestion is slowly getting happening and is finishing up in the small intestine, absorption is also happening in the small intestine. And the water that we take in is actually absorbed. Well, 90%, 80 to 90% of the water we take in is going to be absorbed here, but there's still going to be a lot of water that would be reabsorbed because the chyme, the undigested chyme that gets into the large intestine now, is now going to be processed so that it will be ready for defecation or for elimination. So your fecal matter now, which it was originally your chyme, is now going to be ready and it's going to be excreted in um, in the large intestine at the distal end, right through the from the rectum right through the anus. Okay, that is actually in a nutshell. Like I must have said that in 5 or 10 minutes. So let's take a look at the organs of the digestive system, which falls into two groups. Okay, the first one will be the alimentary canal, the gastrointestinal or the GI tract, all of that particular tract. And the second part will be the accessory organs. So the tract itself is not going to be able to work on the food that it needs to digest without the help of the accessory digestive organs. So your alimentary canal is the continuous muscular tube that runs from the mouth to all the way to the anus. And these this tract is actually the one that digests the food, breaks it down to smaller fragments, absorbs the fragments in right through the lining, uh, dear, right through the lining, and pretty much all the way. And those parts or digestive materials that cannot be absorbed and is actually still in the tract is going to be uh, eliminated through the large intestine. And I mentioned that already. The movement right through that goes right through the digestive system would be the first one that is on the list is actually your peristalsis. It's a major means of propulsion that involves alternating waves of contraction and relaxation. So it is your stomach, rather not stomach, but intestinal um, tubes are lined with muscles, circular muscles that when they contract, they they actually move. They um, they have what you call a pinching effect. So the pinching effect is moving down, and it's actually pretty much like uh, moving the parts along the way as it pinches distal towards the distal end, and that's actually what you call peristalsis. And it is propulsive, and some mixing may occur here. But the mixing of the food primarily is actually not called peristalsis, but it's actually segmentation. Now, what is the difference between peristalsis and segmentation? Peristalsis is actually the movement of food distally. So it is actually the one that's pushing the food down, going towards the large intestine. Segmentation, on the other hand, mixes the food. Segmentation is much like your, let's say, it's much like your dryer. Now, when your clothes, after you've washed them, you put them in the dryer, and 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 hot air is going to be blowing on the wet clothes. You do not expect the clothes just to be receiving the blowing air like that. What the dryer is doing is that it's actually churning the clothes around, throwing it around inside, so that even the middle part of that mix of clothes will receive the hot air so that it can be dry. The dryer is not going to be effectively drying your clothes if the hot air is just blowing on the bunch of clothes. The clothes that needs to be dried will need to be churned around. And the same thing is true with little pockets of food or chyme that is actually in the small intestine. For it to be completely digested, it needs to be churned around so that those enzymes that is produced, the breast border enzymes that is produced by the walls of the intestine can actually reach even the food that is actually right there at the center of the intestine. By the churning movement will mix the food, and that will allow the food in the middle to be picking up the brush border enzymes along the walls of the intestine. So both segmentation and peristalsis work together in order to digest the food. So let's start to look at, now that we know the function of the digestive system, let's try to look at the organization of the digestive system. First of all, let's try to look at the relationship of the digestive organ to the peritoneum. The peritoneum is the membrane of the abdominal cavity that is covering it. It is a serous membrane. You have two types of peritoneum as we know it. We have seen this already in AMP1. The visceral peritoneum is a membrane on the external surface that is covering the digestive organs, or most of the digestive organs, most especially the organs that are inside the abdominal cavity, would have a cover which you call the visceral peritoneum. And right here, the gray material right there, the gray area right there, is the abdominal cavity. And this thing here that is hanging down from the center of the abdominal cavity is actually an organ and is surrounded by a what do you call this, a serous membrane, which you call the visceral peritoneum. The visceral peritoneum is connected to the wall that is also covered by peritoneum, which this time you don't call it visceral because it is not a viscera. A viscera is an organ, and here it's actually a wall. So parietal is actually a word that would mean wall. So the parietal peritoneum is the membrane that would cover the wall of the abdominal cavity. It is connected to the visceral peritoneum that goes around an organ. Most digestive organs are intraperitoneal, which means they are inside the abdominal cavity. They are suspended from the body wall by usually a dorsal mesentery. Now, uh, the the one organ, that organ right there, where you could see my pointer, one organ can actually be connected to another organ, and this organ right here, where that I have my pointer on, is hypothetically something that would be, for instance, hypothetically the liver, let's say, and let's say that this is actually your stomach and or your intestines or your gut, and it's connected to the liver, and the liver is also suspended. It's connected to the what do you call this, the wall of the abdominal cavity. So this particular mesentery happens to be like suspending this organ to the wall, and then this organ to another organ, which is also a mesentery, or at some point some specialized mesentery we are actually going to call, and we will meet them when we get to it, will be the omentum. All right, and then you have an organ that's again suspended into the another part of the wall, which is also another mesentery. Hmm, excuse me. Now, there are some organs that are not suspended inside the abdominal cavity. These organs stay outside or behind the peritoneum. These organs are what you call retroperitoneal. Retroperitoneal. Because they have lost their mesentery during the development, they are not found inside the abdominal cavity. They stayed at the back of the cavity. Therefore, you call that a retroperitoneal organ. What is peritonitis? Well, this is actually an application, clinical application. Peritonitis is inflammation of the peritoneum, which is the cover of the abdominal cavity. It can be caused by piercing the abdominal, piercing an abdominal. It is caused by a piercing abdominal wound, or a perforating ulcer, or a ruptured appendix. The peritoneal coverings would stick together in this case, which would help um, flame up a localized infection. And um, this particular development or inflammatory development can be dangerous and lethal if it is left untreated and it becomes widespread. The treatment for peritonitis, if it's not going to be stopped, can lead into sepsis. So the treatment will be the removal of these debris and mega doses of antibiotics. Like if an appendix would rupture, the ruptured appendix would pretty much like spread a lot of dirt in the abdominal cavity, and it can start an infection right there, and it can cause sepsis. Hey, now we go into something that you will also find this part of the lecture, you will find helpful when you get into the histology of your alimentary tract. All digestive organs have the same four layers. A layer is actually what you call a tunic. You have the four layers of the alimentary tract, and it's made up of the mucosa, the innermost mucosa. Then you have a submucosa, followed by the muscularis externa, which is the muscle layer that makes that gives the gut the ability to move, and of course the serosa, which happens to be the cover itself. Let's take a look at the first layer that I mentioned, which is the mucosa. Take a look at this. That's where your mucosa is, and it points into the middlemost part of um, the cavity of the digestive tube. The, the digestive um, system is actually made up of a tube which starts from your mouth, the opening, the the top opening is opens into the mouth, and all the way to the end, it is still a tube. Even if it's shaped like a stomach, it still is a tube. It goes into the small intestine, the large intestine, all the way down into the rectum and then the anus. And it would still, although the food that used to be looking differently at the end of, or the distal end of the alimentary canal would not anymore look like the food. It, of course, it is, these are undigested material, and it would be your fecal matter that's going to be eliminated on the other end. So it would have the proximal opening, which is the mouth, and the distal most opening, your uh, which is actually your anus. And this entire opening inside, you will have what you call the lumen. The lumen, I'm pretty sure this lumen is going to be part of a question that you will encounter in your lab practical. Please make sure, I know I remember it, that you know that the inside part of the tube of any hollow organ is called a lumen. And the covering of the lumen is actually what you call the mucosa. It is a layer that lines the lumen of its hollow organ. So the function of the mucosa differs from all other parts. First of all, because the mucosa needs to be uh, smooth and should have the ability to smoothly carry the food from the proximal end going towards the distal end and then be able to absorb it as it digests, it must have the ability, because of some specialized cells, to secrete mucus and also digestive enzymes and also hormones. It absorbs end products of digestion also. So the mucosa must have the ability, some specialized cells must have the ability to absorb the tiny molecules which are byproducts of digestion. And it should also have the ability to protect us against infectious diseases because as we go along and digest the food, towards the end of digestion, there will be like the development already. It will be like several hours. It'll take about 4 hours from the food that you eat to the end before it gets to be digested. Imagine if you just ate up sushi, something that is, or even salad, if something that is not been cooked, and you know that there are some microorganisms in there. Your own microorganism that is actually like resident of our gastrointestinal tract has the ability to stop any infection coming from the microorganisms of the food that we have eaten or that's actually part of our diet. And therefore, one of the things that happens there is that uh, it has the ability, this particular mucosa has the ability to protect us from infectious diseases. And that's the reason why we have scattered lymphatic nodules, which is also found in the entire digestive tract. That's what you call the mucosa associated lymphatic tissue, MALT. So mold have several sublayers. The first sublayer of mold is the epithelium, and that epithelium is actually made up of uh, epithelial cells. Let me see. The epithelium is this part here, okay, and that's the part right there, which is, there you go, the mucosa is made up of the epithelium. The second one is the lamina propria. And the third actually is your muscularis mucosa. This is actually the first layer of muscle. Immediately after the mucosa or the lamina propria of the mucosa will be your muscularis mucosa. This is actually that pulls the mucosa into folds. There are some areas in the intestine where you see the inside or the lumen to be thrown into finger-like folds. Okay, in the stomach, you call those folds the rugae. In the small intestine, you call those folds the villi. And so the epithelium, depending on where the epithelium of the mucosa is, the epithelium, let's say, of the esophagus is actually made up of stratified squamous epithelium. Once the epithelium gets into the stomach, the epithelium of the stomach, of the mucosa of the stomach, becomes a single layer only, and it is actually made up of columnar epithelium. And that columnar epithelium uh, would be loaded with goblet cells that would be secreting mucus. And all the way to the small intestine, the lumen gets to be like a simple columnar epithelium, all the way into the large intestine, as a matter of fact. So the only ones that is not lined with simple columnar epithelium will be the esophagus and the mouth, which is actually lined with stratified squamous epithelium. So the mucosa, the epithelium of the gastrointestinal tract, the mucosa, eases food passage. They secrete enzymes and hormones, and pretty much would um, eliminate the mucosa secretions would eliminate friction of food that would be gouging on the walls of the gastrointestinal tract. It makes the passing of the food and the digestion of the food a smooth travel until the undigested food gets to be eliminated. The next layer is called the lamina propria. The lamina propria, if I'm not mistaken, right here, your lamina propria is right after the muscularis mucosa. The lamina propria is this part right there, this part right here. Uh, and I'm looking for how it is out. The lamina propria is made of loose areolar connective tissue. And what the heck is it? Okay, you have the mucosa, the epithelium, you have the muscularis mucosa. Okay, the lamina propria is this layer here that is loaded with, this one here, where I'm passing right through my thing is. It is not beyond the mucosa. It is part of the mucosa. This is the lamina propria. Where is there you go. That lamina propria. Yeah, I'm pointing to the right thing. The lamina propria is made up of loose areolar connective tissue. It's still part of the mucosa, and there's a rich supply of capillaries located here that is needed for nourishment and absorption. It is actually made up of mucosa-associated lymphoid tissue. You see those green spots right there? Those are the lymphoid tissues, and they help defend us against microorganisms that we're eating. They are what you call the MALT, mucosa-associated lymphoid tissue. And finally, the muscularis mucosa, this muscle area. This muscle area produces local movements of the mucosa, and it helps in forming the contour of the mucosa, just like what I mentioned. It pulls the mucosa into shriveling up in order to uh, um, allow a larger surface area of the mucosa. All right. Then we get into the submucosa, which is now deeper, the deeper section of the gastrointestinal tract, deeper than the mucosa. The submucosa is this area here. Okay, this area here, the lighter area here, is your submucosa. The submucosa look like the lamina propria, but the difference between them, the submucosa is loaded with blood vessels and nerves. So they do have a lot of blood vessels, lymphatic vessels, lymphoid follicles, and pretty much the nerve plexuses. This supplies the surrounding GI tract tissues. It has an abundance of elastic tissue that help organ to regain shape and storing large meal. Then finally, you have a thick layer. Actually, it's not a. There you now have thick layers of muscle, which is called the muscularis externa, and I am now in the muscularis externa. The muscularis externa are made up of two layers of muscle, an inner circular. When it says circular, it goes in a circle. The strands or the fibers of the muscle goes in a circle like this. When you cut the muscle along its axis, the circular muscularis circularis is going to look like dots on the microscope. However, if and of course, first let me tell you first about the fibers of the other layer of muscle, which you call the longitudinal layer. So the longitudinal layer the fibers are actually running parallel to the axis of the gastrointestinal tract. So if you cut this longitudinally along its axis, then the circular muscle would look like dots, like this cell, and then the longitudinal muscles would look like fibers that are running straight on the axis. However, if you're going to be given a sample histology slide that would be cut transversely, like it's actually like a rounded sample of, let's say, jejunum or ileum or any of the parts of the small intestine, then your if it is a transverse cut or a cross-sectional cut, then your outer longitudinal muscle would look like dots, and your inner circular muscle, because it runs along the circular um, plane the of the gastrointestinal tract, would look like long fibers. Okay, they would look like long fibers. The inner circular would look like long fibers if it is actually in a cross-sectional cut, and then your longitudinal fibers would look like dots. And these are the ones, this circular layer, uh, pretty much, and the longitudinal layer, in their movement, the circular layer can form pinching. When it contracts, it narrows the opening. And then when the longitudinal layer contracts, it actually moves the food forward or backwards. Of course, forward only, meaning forward going towards the distal end or going towards the anus. And your pinching effect would help pinch out small items or small portions of food so it can be moved towards the, the distal end. So these are the ones that are responsible. These two muscles are the main muscles of peristalsis and segmentation. The outermost layer, the outermost layer. This is the outermost layer, which you call serosa. That's the serosa. Is actually made up of connective tissue layer and the epithelium or the mesothelium. And these are actually the ones that makes up the visceral peritoneum. This is your visceral peritoneum. And they are formed from areolar connective tissue covered with a single layer of squamous epithelium, which you call the mesothelium. In most organs, it is replaced by the fibrous adventitia in the esophagus. So, in an area where, like for instance, the esophagus, where it is not suspended in the abdominal cavity, there won't be any visceral peritoneum because it is not surrounded by any visceral peritoneum because it's not suspended inside the cavity itself. So that particular cover is not called the visceral peritoneum. You call it the fibrous adventitia. Okay. Now, you find this fibrous adventitia or the adventitia uh, in retroperitoneal organs. Uh, But in some retroperitoneal organs that are very close to the wall, you can have also an adventitia and at the same time a serosa or a serous membrane. So we're now going to get into the blood supply and splanchnic circulation. Splanchnic circulation would include the arteries that branches off the aorta to serve the digestive organs. And pretty much the aorta, this is actually the red part, the red portion is actually the abdominal aorta, and the tributaries or the blood vessels that branches off the abdominal aorta, that forms what you call the splanchnic circulation. It includes arteries that branches off the aorta to serve the digestive organs. So you have your hepatic, well, first you have your celiac arteries, the hepatic arteries, the splenic, there you go, the splenic arteries, the left gastric arteries, okay, and the inferior and the superior mesenteric arteries, and the hepatic portal. So, what is the difference between the hepatic arteries and the hepatic portal? Well, the hepatic okay, food that you have, food that we have digested, pretty much is going to be um, delivered into um, into circulation and would need to go and get into the liver. Food is now going to be delivered from the venous system into the portal vein and into the liver. From the liver, the food now is going to be processed. And there's so many things. The liver is one of the best organs, life-saving organs that we have, because it is the one that would be filtering the food of any toxic substances that could kill us. And therefore, it breaks down these toxins. And any of the food materials that we have digested so far that we need to convert into our own what do you call this, into our own human uh, human human molecules pretty much is done by the liver. Let me just give you a very brief example. Now, if we are very fond of eating chicken breast because pretty much that's the only thing that's our favorite and that's the only thing we want to eat, that doesn't mean that we're going to turn our muscles will turn into chicken breast muscles. No, our body will process the chicken breast, will break down the chicken breast into its simplest form, the amino acids, and then your own body will not anymore recognize it as chicken breast. These amino acids are now going to be formed into the protein that is actually human protein. So it's now going to be processed in the liver into basic macromolecules that is going to be distributed to the different cells and is going to be forming our own human muscles or part of our blood or whichever part of our system. The amino acids are actually it makes up the human protein. So we do it right here in the liver, but from the gastrointestinal tract, it is going to be delivered right through the what it calls the hepatic portal system, through the hepatic portal vein, right there into the liver. Then from the liver, as soon as it's actually finished, it's now going to be delivered into the different parts of the body through the hepatic arteries. So that's actually part of what you call the hepatic portal circulation. It drains nutrient-rich blood from the digestive organ, delivers the blood to the liver for processing. And so let's now try to look at, or let's look at now the control of the digestive system. Your digestive system has what you call enteric nervous system. The enteric neurons make up the bulk of two main interconnecting intrinsic nerve plexuses. We have what you call the submucosal nerve plexuses, okay, and your myenteric nerve plexuses. So GI tract has its own nerve system, has its own nervous system. You call this nervous system the enteric nervous system. It is also called the gut brain. Contains more of the neurons. It actually contains more neurons than the neurons we have in the spinal cord. The gut brain is made up of enteric neurons that communicate extensively with each other. The major nerve supply to the gastrointestinal tract wall that pretty much controls the movement of food inside the gastrointestinal tract. And this particular group of neurons is actually within a particular cluster of nerve, which you call the myenteric nerve plexus. So you have two of them interconnecting nerve plexuses. The first one is what you call the submucosal nerve plexuses, which is actually the one regulating glands and smooth muscles in the mucosa itself. And then you have the myenteric nerve plexuses, which controls the gastrointestinal tract motility. And this enteric nerve system participates in both the long as well as the short reflex arcs of your gastrointestinal tract. So what is the short and the long? I mean, why short and why long? The short reflexes is mediated by the enteric plexuses, the gut brain, and responds to stimuli in the gastrointestinal tract. The long reflexes respond to stimuli arising inside or outside of the gut, such as from the autonomic nervous system. Now, the autonomic nervous system that controls our gastrointestinal tract is coming from the parasympathetic nervous system. This parasympathetic nervous system enhances the digestive process. And I'm pretty sure you still remember this from the autonomic nervous system that you have read and studied in AMP1. So parasympathetic system enhances the digestive process, and the sympathetic would inhibit digestion. So let me get an example. the sympathetic nervous system is the sympathetic nervous system is actually the system that is controlling us when we are in a fight, fright, flight response. So when we are excited or when we are um, um, stressed out, we are under the sympathetic response. And under that time, while we are excited, we're in love, or we are under extreme emotional tension, we are not going to be hungry. We don't have any appetite to eat, right? Now, when we are relaxed and if we're just watching TV, that's when we will feel hungry. So when we are relaxed, the parasympathetic is actually the one that is controlling us. And this is actually the one that really makes us go and capable of handling the um, the workmanship that is involved in the parasympathetic nervous system or in the digestive process. The digestive process is parasympathetic. And that's why if we want to eat and we would like to digest the food that we ate, after eating itself, we tend to be a little bit woozy. All of our blood is actually now in the enteric nervous system, and pretty much this particular um, the blood that's pulling around the enteric nervous system is helping in the movement of the gastrointestinal tract, and where the aim is really to digest the food that we just ate. Okay. Okay, let's take a look at the neural reflex pathways initiated by stimuli inside or outside the gastrointestinal tract. Three concepts, three concepts regulate gastrointestinal activity. Digestive activity is provoked by a range of mechanical and chemical stimuli like receptors, okay, the external stimuli such as receptors that is located in the walls of the GI tract organs, and they respond to stretches and changes in the chemical nature of the environment and the pH and the presence of substrate and end products of digestion. Pretty much these external stimuli uh, are the ones that is acting as what do you call this, a stimulus so that our our gastrointestinal tract will move, and where it's going to be or in which direction it's going to be going depends on the stimulus itself. All right, so if we are eating food that is high in protein, then the behavior of the gastrointestinal tract will be to secrete um, substances, chemicals, hormones that would lead into the direction of uh, digesting the protein that we just ate. All right. It is not going to be going towards the direction of a protein if we ate a lot of carbohydrates. If we ate a lot of carbohydrates, then it will be a different set of chemicals that will be released in order that these carbohydrates will be digested. But of course, it doesn't happen that way because the food that we eat is always a mixture of almost like anything that we would like to put in our mouth. You know, even a dish that we're actually eating would have protein in it, would have fat in it, would have carbohydrates in it, would have fibers in it, and therefore it will um, open up an orchestra or of a mixture of substances that coordinates with one another in order to digest uh, one food that is made up of a mixture of substances. So that food that is going to be digested is going to be digested with the help of reflexes that is originating from the food right there, and would send stimulus and would reach the brain, and the brain with the central nervous system will now pretty much like send off a stimulus wherever whatever it is supposed to be doing, um, that would affect or that would cause the movement of the gastrointestinal tract to secrete substances or chemoreceptors or almoreceptors in order to uh, make the gastrointestinal tract move in a way where those substances can be digested. Let's take a look at the functional anatomy of the digestive system. Start from the mouth. Let us start from the mouth. The mouth is where the food is chewed. That's why we have our teeth, and mixed around and would pretty much like commence the digestion with the help of enzyme containing saliva. This is actually the one that starts the process of digestion. And once saliva gets in touch with or gets mixed with the food, you don't call it any more food, you call it bolus. So the bolus now is going to be swallowed, and the process is initiated where your saliva is actually formed, which is in the mouth. And with the help of the tongue, the salivary glands, the teeth, all of these organs that is found in the buccal cavity pretty much is the one that is responsible for the initial digestion of food. I am holding you responsible for knowing pretty much uh, this particular sagittal section of the model of the head in the lab, just to make sure that you know the parts. For instance, your hard palate right here is this one here. Then behind the hard palate is actually your soft palate. Then you have your uvula. What is the function of the uvula? The uvula, when you swallow food, folds up right here. This is like a ding dong bell, right? In the cartoons, we see that as a ding dong bell. When you swallow food, it folds up, and it's going to cover this opening to the what do you call this? The the nasal cavity. It will prevent the food that you are eating from being thrown into your nasal cavity. That's why you do not talk. This is one of the reason only why you are not supposed to be talking while you are eating and swallowing. So when you want to talk on a dinner table, you don't do it when you have food in your mouth. You first swallow the food in your mouth, and then you can talk. You can discuss things in the dinner table. And that's going to make sure that because air moves here in your from your oral cavity or in your nasal cavity and down into the voice box, where it's actually the pathway that needs to be like uh, going through for you to talk, right? And when food is here, the food can actually be dislodged anywhere. It can go up there and you cough it out, or it can go down here in the trachea, because you're going to also disrupt the epiglottis from doing its job, and therefore your food can actually accidentally get in there right in the trachea, and you need to cough it out also. So in order to avoid those embarrassing uh, mistakes, then you'll have to make sure that you swallow first your food, you drink your water, you swallow your food, then you talk. You do not talk while you have food. That's where, well, a lot of people do that, okay? And then get into, some people really are very good at doing that and would not get into an embarrassing situation. But if you're not used to that, make sure that you just eat first and then you discuss. So, the walls of the mouth is lined with stratified squamous epithelium. And these are tough cells that would resist uh, abrasion. The cells of the gum, the hard palate, and part of the tongue are actually keratinized for extra protection. The lips and the cheeks is composed mainly of fleshy orbicularis oris muscle. Orbicularis oris. This is actually the muscle that is called commonly known to be the kissing muscle, orbicularis oris. And the winking muscle is the orbicularis oculi. Now the cheeks, this part here will be your cheeks. They are composed of the buccinator or the buccinator muscles, the oral vestibule. These are the recesses internal to the lips and cheeks and external to the teeth and gums. The oral cavity proper, this is the oral cavity proper, lies within the teeth and the gums. The labial frenulum, which is, well, that's actually the labial frenulum. That's a labial frenulum. This is your inferior labial frenulum, and you have another labial frenulum right there, superior labial frenulum. These are median attachment of each lip to the gum. The palate forms the roof for the mouth and has two distinct parts, and I mentioned that already, the hard palate and the soft palate that ends into the uvula. The palatine tonsils. The palatine tonsils are located in the fauces. Uh, Let me see. Can we find the palatine? There you go. Palatine tonsil. The white thing there is your palatine tonsil right there. This two. Okay. And that's actually the uvula. Finger-like projection that faces downward from the free edge of the soft palate. It prevents the food from uh, going up in your nasal cavity as you are swallowing. The tongue, good old reliable tongue, occupies the floor of the mouth. It is composed of interlacing bundles of skeletal muscle. Yes, it is pure red meat. Okay, the function of the tongue very much will be for the digestive digestive system will be to grip, reposition, and mix the food during chewing. It is actually used in the formation of bolus, which is a mixture of food and saliva. It is helping initiate swallowing. It is also a big help in speech and taste. The intrinsic muscles change the shape of tongue, and the extrinsic muscle will be altering the tongue's position. The lingual frenulum is the attachment of the tongue on the mouth or the floor of the mouth. Now, on the superior surface, this is the superior surface of the tongue, you would have like papillae and peg-like projections of underlying mucosa. And you have those finger-like projections, okay, are what you call the papilla. And you have different papilla in what do you call this, in the surface of your tongue. The filiform papilla are fine finger-like projections that gives the tongue roughness to provide friction. And only the only one that do not the filiform papilla is the only one type of papilla that do not contain taste buds. It gives the tongue a whitish appearance. Then you have the fungiform papilla or papillae. These fungiform papillae, and usually they are bigger, those bigger dots right there. They are mushroom shaped and they are scattered widely over the tongue, and pretty much they cause the reddish appearance of the tongue. These would have already like a good amount of taste buds in it. Then you have the circumvallate, which is the biggest of all the uh, papilla of the tongue. Right here, in this particular area here, right here, you will have a good number of circumvallate papilla. These are the circumvallate papilla. Let me see. There is something circumvallate papilla. I wonder why. There. The valid papilla, the V shape. There you go. Those circumvallate papilla are actually the biggest of all the papilla, and they do have also taste buds. The foliate papilla is located on the lateral aspect. There you go, on the lateral aspect. There you go, of the tongue on the posterior edge, and pretty much is very active and is uh, quite not so formed when we are young. That's why there are so many things when we are young, there are so many things in the food that children do not appreciate because certain parts of their taste buds are not capable of really tasting yet. So their taste buds are really bland. They don't want anything that you put too much salt, too much sweetness, too much of anything for a child they would not want. So you give them something that's really natural, and even if you think it doesn't taste anything, that's actually what's good for them. It's because their taste buds cannot appreciate yet those strong, powerful taste of the adult food. Okay, here's a homeostatic imbalance, which is pretty much like uh, the lingual frenulum. The lingual frenulum, like this one here, is that particular uh, connect. It is that particular membrane that connects the tongue to the floor. This is a normal lingual frenulum. This is a moderate lingual frenulum. This is a severely short lingual frenulum. When you have a severely short lingual frenulum, you're going to have a hard time move your tongue so you can speak better. Children whose problem really is speech when they're growing up is that might have problems with the length of their lingual frenulum. Sometimes severing that lingual frenulum up to a certain point will help in the movement of their tongue and will help improve their vocabulary and their speaking after the operation. When you have a severely short lingual frenulum, this is actually the situation where the child is called tongue-tied or fused tongue. The restricted tongue movement will distort speech, and the treatment will be to surgically snip the lingual frenulum. I know of a child who had a problem like this. I know of a man who had the problem like this, and so the parents pretty much like resorted to approving the doctor's suggestion to snip the lingual frenulum. And guess what? Now he is probably the most talkative man, but when he was a child, he barely talked because he was tongue tied. So now we get into the saliva. The saliva is produced by salivary glands that is surrounding the mouth. You have the parotid gland. You have also the submandibular, which is under the mandible, under the tongue and under the mandible. Then you have the submandibular, which is pretty much like there. This is the one that is really under the tongue. We call the submandibular. Oh, sorry. That is the submandibular gland, sublingual gland, and the submandibular gland is under the mandible. Okay. and this is the parotid gland. So what is the function of the saliva? What is it? Why is it so important? Well, depending on what the chemicals are in the saliva, depending on what the chemicals are in a liquid, pretty much, and you would know what the character is or the nature of that liquid is. And now we're talking about the liquid in the mouth, in our mouth, which we call saliva. It cleanses the mouth. It dissolves food chemicals for tasting. It moistens the food. It pretty much helps compact the food into bolus. It begins the breakdown of starch with the enzyme amylase. We have salivary amylase in the mouth, and most saliva is produced by the major salivary glands, which I mentioned already, outside the oral cavity, and then some a lot of scattered minor salivary glands through are actually located throughout the oral cavity. They pretty much like um, slightly augment the function of um, the saliva on food. The major salivary glands are your parotid gland, which is anterior to the ear, anterior to the ear, and external to the masseter muscle. This is your masseter. The parotid duct opens into the oral vestibule next to the second upper molar. The submandibular gland is medial to the body of the mandible. Duct opens at the base of the lingual frenulum. Then you have the that's a sublingual gland under the tongue and opens via about 10 to 12 ducts into the floor of the mouth. Now, when would be in a 24-hour cycle of our mouth? When is actually the time that we feel our mouth would be smelling the worst? Even if we brush our teeth like twice or three times a day, that particular time of the day, you think that your mouth is actually smelling the worst, will be your early morning breath. Why? Why the early morning breath? Even if you brushed your teeth, you pretty much cleaned around your teeth the night before, and you are taking care of your teeth and your mouth, still early in the morning, that is the worst part of the day for your mouth. It's because the entire time you are sleeping and the secretions of your salivary gland is actually slowing down because you are not moving your mouth and you are not actually making your salivary glands secrete saliva. So a lot of the bacteria that is already right there in your mouth, no matter how much you cleaned it the night before, is going to multiply and will make your saliva, whatever is there that's hiding as you are sleeping, it will actually smell in early in the morning. But the moment you start talking and then you start producing new sets of saliva and then you just swallow in or you just throw out the old one, then your breath would not be smelling as bad anymore until of course you you wanted to really clean it by brushing your teeth early in the morning. But the movement of your mouth when you're chewing gum, when you're chewing something, would excite and stimulate your glands to produce saliva, and this saliva is the one that makes your breath really smells better. Okay, now this is histology, the histology part of the salivary gland. I had to look for this special picture, okay, just to let you know that salivary glands are composed of two types of secretory cells. The first type is what you call the serous cells. The serous cells makes up the serous acini. They produce watery secretion and they have a lot of enzymes, ions, and a little bit of mucus. but they are really very watery. Then you have your mucus cells. The mucus cells form what you call the mucus acini, and the mucus acini produce mucus. So take a look at this and pretty much like tell me which one is actually the serous acinus and which one is your mucus acinus. Well, make sure that you know that the serous acinus would cover around the duct like so, looking like a cap. And these are actually your serous acinus. I took this picture and make sure that this is actually will what you will see. I want you to know that the acini would have dark purple staining cells. Meanwhile, your mucus acinus would have translucent cells, and these are your mucus acinus because they are producing mucin or mucus. So these will be the mucus producing cells, which makes up the mucus acini, and this will be your water, the part of the saliva that would be producing watery secretions, which you call the serous cells. The parotid and submandibular glands contain mostly serous cells, but the sublingual gland consists mostly of mucus glands. So you have to make sure in your exam, I want you to know that in your exams, in all of our exams that would have pictures of histological pictures or diagrams, all of the histological pictures are labeled exactly what they are. So we are not going to give this to you without letting you know that this particular picture is actually a mixture, is actually like uh, maybe that I must have pushed it all the way in and so I cannot see the picture. So this must be a submandibular gland because it has both serous and mucus. And if I have a parotid gland or a sublingual gland, then I would have really a picture that would have mostly mucus cells. But because I have a mixture of both, then it could be parotid or submandibular gland because both parotid gland and submandibular gland contain mostly serous cells. Okay, submandibular will have a mixture of both. I guess this is submandibular. Parotid will have a lot of the purple uh, serous cells, but your sublingual gland contains mostly the mucus cells. So, salivary glands again. Let's take a look at control of salivation. We produce about 1,500 ml. That is actually like a liter and a half a day, okay, of saliva. We can produce 1,500 ml of saliva. Minor glands continuously keep the mouth moist. Major salivary glands are actively activated, right, by parasympathetic nervous system. And a strong sympathetic stimulation would inhibit salivation and would cause dry mouth or xerostomia of the mouth. The teeth. You're going to have pictures, diagrams of the teeth also in the lab, and you will have to know and have to identify the parts of the teeth. You're responsible for knowing that. Okay. The teeth lie in sockets that is in the bone, which we call the alveolar sac of the bone, and teeth lies in these alveolar sockets or pockets. It is used for chewing or mastication. And your dentition usually have formula. You have to memorize the formula. The primary dentition or the primary teeth, the baby teeth, the ones that we have before we lose them initially, will be we have 20 milk teeth or 20 of your deciduous teeth. Then once they all like um, once they all gets pulled out, okay, we lose them. They're going to be replaced by 32 deep lying permanent teeth. And these permanent teeth are made up of the four types of teeth that we have. We have incisors, canines, premolars, and molars. Now, your 20 deciduous teeth uh, pretty much would not have, the deciduous teeth would not have molars. You have premolars, canines, and incisors, but not molars. No, it will not have premolars, it will have molars. I mistake. Okay, so this is actually the formula for your uh, baby teeth. You have uh, on the top, you have pretty much two incisors, one canine, two molar on the upper jaw. On the lower jaw, you have two incisors, one canine, also two molars, a total of 20 teeth. On the permanent teeth, you will have the appearance of, you still have your incisors, canines, and molars, but you will have the appearance of two more premolars on both the upper teeth and the lower teeth. You're responsible for knowing also the parts of a teeth. Here I'm showing you the longitudinal section of the canine tooth within its bony socket, which we call the alveolus. Each tooth has several structures. Okay, you have to make sure that you know them. This part here that is actually exposed, okay, is the crown, the exposed part above the gum, part of the teeth that you see when you smile, or the ones that you want to stain whiter if it's actually getting discolored already because of age. Then the one that is below the gum line, right there, this part here, is actually your root. It's a portion embedded in the jawbone. So this is actually your root. There you go. Then the neck actually is that portion that connects the crown and the root. The cement is actually the calcified connective tissue. And this is actually the cement right there, the calcified connective tissue. Okay, the cement is actually between the dentine of the teeth and the periodontal ligament. So you have a cover right there which called the cement that is calcified, but it is connective tissue. The periodontal ligament, this pink band right there, is actually the one that forms the fibrous joint. Then you have your uh, gingival sulcus. This gingival sulcus right here, right there, is actually the groove. The dentine is this blue section right there. The dentine is actually the bone-like material that is under the enamel. You have your pulp cavity, and the pulp cavity is right there in the middle, which is surrounded by the dentin. And your pulp is the connective tissue, blood vessels, and nerves that is inside the pulp cavity. The root canal is mm, the root canal right there. The root canal, okay, pretty much as a the as a pulp cavity extends into the root. That's actually the canal right there that helps uh, the elements of the pulp cavity, blood vessels and nerve would get into to join in the blood vessels and nerves outside of the or into the bony tissue. Then the apical foramen, the hole right there, it's actually where the rest of the nerves and blood vessels would have to go through. Okay, so these are some of the homeostatic or clinical aspects of uh, teeth, the bad parts of the teeth. Uh, okay. The first one is actually impacted tooth. That's actually impacted tooth. A tooth that remains trapped in the jawbone is actually an impacted tooth. Can cause a good deal of pressure and pain. Wisdom teeth are most commonly involved here. And actually, the one that gets to be impacted in most people who would have this problem will be the wisdom teeth. The treatment will be just pull out the wisdom teeth, surgical removal of the impacted tooth. Then you have dental caries. Oh, cavities. Oh, a lot of them would have cavities. Oh, that is terrible. This is actually the demineralization of the enamel and dentin from because of bacterial action. Dental plaque is actually the development of a film of sugar, bacteria, and debris that adhere to the teeth. And you could see some teeth here, oh, that has plaque. That adhere to the teeth. The acid from bacteria would dissolve the calcium salt, and then the proteolytic enzyme and organic matter will act on it. The prevention of this is actually to pretty much like clean your teeth daily, more than once daily, daily flossing and brushing. And you have also gingivitis. This is actually a plaque that calcifies to form calculus or tartar. It disrupts the seal between the gingivae and the teeth. This is when anaerobic bacteria would infect the gums. Infection here is reversible if you can actually have the calculus removed. Now, the only thing you need here is a visit from your a visit to your dentist. Periodontal disease. Periodontal disease or periodontitis pretty much is neglected gingivitis, which can escalate to a disease. Here, the immune cells attack not only the bacterial intruders but also the tissues. It can destroy periodontal ligament and can activate the osteoclast, which is actually happening to be it's a white blood cell, okay, that leads to the dissolving of bone and possible tooth loss. So the risk factors here usually is smoking or diabetes and pretty much oral piercings. That could really be dangerous. I will see you in the second um, video of the digestive system. Bye for now.